some pictures. So, mitochondria is very important in cell injury, both in necrosis and apoptosis. So, mitochondrial damage can cause decreased ATP generation and also leakage of pro-apoptotic proteins. So, mitochondria are important by leakage of pro-apoptotic proteins in apoptosis and by lack of generation of ATP in necrosis. Also calcium, entry of calcium means influx of calcium inside the cell has adverse effects. Activation of multiple cellular enzymes and also increases mitochondrial permeability. Different enzymes, endonucleases, phospholipases, that they can degenerate proteins, lipids, so phospholipids including membranes. ROS means reactive oxygen species. They are free radicals. Free radicals damage everything, lipids, proteins, and DNA. And membrane damage is also important in cell injury, including plasma membrane and lysosomal membranes. All the membranes in organelles, plasma membrane, loss of cellular components, and lysosomal membrane, which lysosomes contain many hydrolytic enzymes. So these enzymes can leak out of the membrane and cause degeneration of organelles, other materials inside the cell. And protein misfolding and damage due to mutations, due to irradiation, it can activate pro-apoptotic proteins. So usually the outcome of protein misfolding and DNA damage is apoptosis, not necrosis. So pay attention that the result of these injuries is not always necrosis, not always apoptosis. So usually the result of reactive oxygen species, the entry of calcium, is necrosis, but they can also be apoptosis. And if the damage to protein and the DNA is mild, usually it results in apoptosis. But when the damage is very severe, it will go for necrosis. And as I said, mitochondria are both important in apoptosis and necrosis. So some of the figures in Robin's textbook are very important and can give us a lot of information. So these are the elements that change are affected in a cell injury. And this picture, which is also very important, shows that ischemia means presence of an obstruction inside the vessel causes lack of blood flow distally, and it causes first of all, it affects mitochondria, causes lack of oxidative phosphorylation, because mitochondria can do this metabolic pathway by using oxygen. So when there is a lack of oxidative phosphorylation, there will be low ATP, and these results are again important. When there is no oxygen to do the oxidative phosphorylation, then anaerobic glycolysis is performed. Anaerobic glycolysis means the changing of glycogen or glucose to lactic acid and then pyruvic acid. So lactic acid is increased, glycogen is consumed, it is decreased. Lactic acid is increased, and so acidity inside the cell increases. And this increased acidity inside the cell causes clumping of nuclear chromatin. So one of the effects of lack of oxidative phosphorylation due to hypoxia, which the most common cause of which is ischemia, is presence of anaerobic glycolysis as a compensation. And another effect of lack of ATP is its effect on sodium pump. This sodium-potassium pump doesn't function normally. There will be entry of calcium Normally, the concentration of calcium inside the cell is very low. It is kept at low concentrations. So when this pump doesn't function normally, there will be influx of calcium and sodium, and due to osmosis rule, water will also accumulate inside the cell, and also this will cause swelling of the cell and all the organelles inside the cell. So endoplasmic reticulum swelling, cellular swelling, and this swelling will cause loss of microvilli and presence of blebs. And when sodium comes inside with calcium, there will be efflux of potassium. Potassium efflux, sodium influx. So this is important, that swelling of the cell is the first manifestation of necrosis. The first morphologic manifestation of necrosis is cell swelling. But as you know, in apoptosis, the cell is changed to what? Do we see cell swelling?
No, shrinking.
In apoptosis, we see shrinkage of cells. So cell swelling is as the first manifestation, morphologic manifestation of necrosis due to this change of the sodium pump. And another effect of lack of ATP is detachment of ribosomes, because ATP is important for lipogenesis, protein synthesis. So when there is low ATP, there will be low protein synthesis, and this lack of protein synthesis will cause lipid deposition. So these three effects are seen due to lack of ATP. Again, this picture is very important. That we can make some questions with these effects, that we can say that which one is correct in cell injury. So we should know these changes. And mitochondria act in two ways in necrosis and apoptosis. As I said, mitochondria have two membranes, outer membrane and inner membrane. And the inner membrane has proteins. On this inner membrane, there are proteins for doing the oxidative phosphorylation. So, in necrosis, there are some proteins on this inner membrane, such as cyclophilin D. There are proteins on the inner membrane, such as cyclophilin D, that when there is lack of ATP production, they make a channel-like structure. So this protein is normally closed on the inner membrane of mitochondria. This is the mitochondria, this is the inner membrane, outer, inner. Some proteins here are present, such as cyclophilin D, that due to injury, lack of ATP, this makes a channel-like structure, and protons come out of the mitochondria, and this causes again lack of ATP generation. This loss of potential inside the mitochondria goes for necrosis. So, and this part is called mitochondrial permeability transition pores. When they cause channel-like structures, they are called pores that normally we don't have. When there is dysfunction of these proteins, these make pores or channel-like structures. So this inner membrane is functioning in necrosis. So H plus has way out of the mitochondria. There is loss of membrane potential, inability to generate ATP, and goes for necrosis. But in apoptosis, the mechanism is different in mitochondria. There are some proteins in the intermembranous space of mitochondria, the most important of which is cytochrome C. These cytochrome C proteins. Again, we should be aware of the presence of some apoptotic proteins. So those cyclophilin proteins were on the inner membrane. Here there are some other proteins of the BCL2 group of proteins. These are around 20 types of proteins. Some of these are cause apoptosis, pro-apoptotic. Some are anti-apoptotic, and some are sensors, such as BACs and BAC proteins. They cause apoptosis. So in injury, these BACs and BAC proteins of the BCL2 family, which are pro-apoptotic, they make a channel-like structure, and this cytochrome C comes out of the mitochondria. And this cytochrome C activates caspases, the main enzymes functioning in apoptosis. So these cytochrome C and other pro-apoptotic proteins have way out of the mitochondria and do apoptosis. So mitochondria are important in both pathways. Now I explained the mechanisms of apoptosis. This is a cell. This is the mitochondria. And this is the membrane. We have two ways in apoptosis. One is mitochondrial or intrinsic pathway. The other is extrinsic or death receptor pathway. In the mitochondrial pathway, I said that there are proteins of apoptotic type that they make channel-like structure, and those cytochrome C proteins have way outside of the mitochondrium. Now when they come out, they activate another protein called APAF1, apoptosis activating factor 1. So this is called, this is a multimer. This cytochrome C is attached to APAF1 and makes apoptosome. So cytochrome C comes out of the mitochondria, activates APAF1, making the complex apoptosome. This apoptosome activates an enzyme called caspase 9. There are two types of caspases. Initiator, including this caspase 8, initiator, and then they activate another type of caspases called executioner caspases, such as caspase 3 and 6. Caspase means C means that it has cysteine and it cleaves the protein after aspartic acid. So this abbreviation caspase is proteins containing cysteine that cleave the protein after aspartic acid. There are two types, initiator types of caspases, executioner types of caspases. Executioner types of caspases are common in both mitochondrial pathway and the extrinsic pathway. They are the same in this, but they are different in initiator ones. So these caspases are enzymes that can degenerate nucleus. These enzymes, caspases, are the most important enzymes in apoptosis. They are the enzymes that make apoptotic bodies. As we know, in apoptosis, the cell membrane is intact, but we have formation of some apoptotic bodies due to these caspases. These apoptotic bodies that can contain some of the organelles of the cell, they separate from the cell and are rapidly phagocytosed by macrophages. So these apoptotic bodies are rapidly phagocytosed by macrophages. This is phagocyte, for example, a macrophage. And as I said before, because this phagocytosis is done rapidly in apoptosis, there is no inflammation. So one of the differences between necrosis and apoptosis is lack of inflammation in apoptosis due to rapid phagocytosis of apoptotic bodies. So in the intrinsic or mitochondrial pathway of apoptosis, this cytochrome C coming out of the mitochondria causes activation of APAF1 making apoptosome. It causes activation of initiator caspase, which is 9 in intrinsic pathway, and then it causes activation of another type of caspases called executioner caspases, which is common in both intrinsic and extrinsic type of apoptosis, such as caspase 3 and 6. And this causes the production of apoptotic bodies. Now the extrinsic pathway. So this was for the mitochondria, which is more common. Mitochondrial pathway of apoptosis is more common than extrinsic or death receptor pathway. Now what is the death receptor pathway or extrinsic type of apoptosis? There are some receptors on different types of cells called death receptors, such as FAS, such as TNFR1. These are TNF type of receptors, tumor necrosis factor. Tumor necrosis factor. Both FAS and TNF receptor type 1 receptors are death receptors. Normally, there are some growth factors that they are combined with receptors on the cell and cause growth of the cell, and signaling pathways are activated to survive the cell. But if there is lack of the growth factors important for cell growing, this death receptor is activated by attachment of another type of molecule called, for example, FAS ligand, which is present on T cells. T cells are some of the lymphocytes We have B cells, T cells, natural killer cells. So one type of lymphocyte is T cell. Cytotoxic T cells. And T cells are divided into helper T cells and cytotoxic T cells. These cytotoxic T cells can directly kill infected cells. These T cells have a ligand on them called FAS ligand. This FAS ligand can bind this FAS and activate this death receptor pathway. Attached to this receptor on inside of the cell, there are some proteins called FAD. FAS-associated adapter protein, FAD. When this is activated, also another FAS receptors bind together, make a complex. Here the FAD is activated. By activation of these FAD proteins, then another type of initiator caspases are activated, such as caspase 8 or 10. This causes activation of the same type of executioner caspase. This is, so as I said, initiator caspases are different, but for both pathways, the executioner caspases are the same. It can be caspase 3 or 6. Then it causes the same thing, the same actions. So this is the extrinsic or death receptor pathway. That receptors present on the cell membrane, activating this FAD complex, activating of the initiator caspases of other type than mitochondrial, and then doing apoptosis. So this is the two pathways in apoptosis. Yes.
Can you repeat it?
We have two pathways in apoptosis, mitochondrial or intrinsic, and cell death receptor or extrinsic. So the injurious agent can affect the mitochondria, which is the most common type, with lack of oxygen, toxins, chemicals, drugs, whatever causes injury to the mitochondrion, of which type of the has effect on the outer membrane of the mitochondrion that activates apoptotic proteins. Apoptotic proteins on the outer membrane that I said are the BCL2 family. Apoptotic proteins are, for example, BAX and BAC. These proteins are activated. I said we have also anti-apoptotic proteins. For example, BCL2, BCLXL, MCL1. These are anti-apoptotic proteins. If these proteins are activated, it prevents apoptosis. And again, we have sensors that regulate both anti-apoptotic and apoptotic proteins, including BAD, proteins like this. These are sensors. These apoptotic proteins of the BAX and BAC type are activated. They make a channel-like structure. Cytochrome C comes out, activates APAP1, then activates caspases. But in extrinsic pathway, it's different. It affects death receptors of the TNF family, including FAS and TNFR1. by attachment of ligands such as Fas ligand. This pathway is activated inside the cell, and attached to that receptor there are some other proteins called FAD. By activation, they activate initiator caspases of other type 8 and 10. 10 is in humans, 8 in animals. This activates executioner caspases, which is the same in both pathways, 3 or 6, and this causes apoptosis. So two pathways of apoptosis, but we have examples, some examples that in these examples mitochondrial apoptosis is seen, and we have some examples that extrinsic pathway of apoptosis is seen. For example, if there is an infection, and the duty of cytotoxic T cells is to kill that infected cells. So due to infections, if cytotoxic T cells are activated and want to kill the infected cell, they use their Fas ligand and kill the cell through extrinsic death receptor. So killing of the cell through apoptosis by the cytotoxic T cells is by the extrinsic pathway. And again, normally, physiologically, we have the thymus gland, which is the place of maturation of our T cells. These T cells are not always normal. And sometimes we have T cells that have abnormal antigens on their surface. Some of them are immature and some of them are dangerous for other cells. These cells should be deleted. And again, this deletion is done through apoptosis. In this apoptosis, if there are abnormal antigens, its deletion is done through the death receptor pathway. But if there are two things here. There are some, again, T cells that if there is no antigen to stimulate them, those T cells that don't see or feel any antigen to be activated, they should also die. But this type of death of lymphocyte is through the mitochondrial pathway. So there are some delicate points here that if we have T cells of the abnormal type, the dead cell is through death receptor pathway. But if if there is no antigen to stimulate T cells should die. There is no antigen. So this type of death is through the mitochondrial pathway. Again, if in most of our organs, the growth, the physiologic reaction of the cells is through secretion of hormones. So if there is lack of hormone secretion, the cells should die through apoptosis, and this type of apoptosis again is of the mitochondrial type of pathway. So if you look at the Robbins textbook and also the slides that I have given you, you can find some examples like this, and these can come in questions that in which of the following, for example, the type of apoptosis is through the mitochondrial pathway or the other. So apoptosis has these mechanisms. I told you last time the reperfusion injury, yes, that if that ischemia is treated, then the type of injury is more severe due to oxidative stress, due to inflammation, due to more calcium that comes through the new blood that is flowing there normally, and activation of the complement 6 system. And did I tell you the toxic injury, chemical toxic injury, directly and indirectly? Mercury, yes. So I told you some examples of necrosis, and apoptosis, so we can have it in DNA damage, in accumulation of misfolded proteins, and again, cell death in certain infections is through apoptosis, such as in viral hepatitis, the type of cell death is apoptosis. In viral hepatitis, some cells die due to the presence of virus. Some of the hepatocytes are lost. So, imagine these are hepatocytes. Hepatocytes are plump cells, and they have nuclei, a lot of cytoplasm, and it's eosinophilic. In viral hepatitis, some of these cells are lost. There is loss of cell. And this type of cell loss in viral hepatitis is not necrosis, is apoptosis. So these lost cells that first they shrink and make apoptotic pycnosis of the nuclei, the cell shrinks and pycnotic nuclei and then disappears by caspases. So the type of cell death in viral hepatitis is apoptosis. Because again, in some exams, they like to give this question that if this type of cell death is necrosis or apoptosis, is it apoptosis? But here in viral hepatitis, is the virus the cause of cell death or not? Is the virus itself the cause of cell death? Yes? Yes, for viral hepatitis, for some viruses, not all. In viral hepatitis, the cause of apoptosis is not directly due to the virus, but due to the cytotoxic T cells. So this is the immune response that by reaction of these cells causes apoptosis, and so it will be apoptosis of the dead cell receptor pathway extrinsic by that Fas ligand causes apoptosis. But some other viruses such as adenoviruses again cause cell death by apoptosis. But this time the cause of apoptosis is directly due to the virus itself. So some viruses cause directly and some indirectly through immune reaction. Again, this is also true for diabetes mellitus. In diabetes mellitus, again, we have type 1 and type 2. In type 2, there is loss of beta cells. And this loss of beta cells, again, is due to the immune response of body, due to the inflammation that is present around the islets. So this is some examples for apoptosis.
Can you have more examples?
More examples for what?
Apoptosis and necrosis.
Apoptosis can be physiologic or pathologic, as maybe I have told you. Apoptosis in embryogenesis is physiologic. Some cells develop and the previous cells die through apoptosis. Maybe I told you that if we don't have apoptosis through embryonic period, our fingers couldn't separate. This is a type of apoptosis. Or normally, for renewing of cells, again, in adults, in skin, in intestinal epithelial cells, cells should die for having newer cells. This is apoptosis. And again, physiologically, the presence of immature and dangerous cells if they die, again is through apoptosis. So these are examples as physiologic. And pathologic atrophy is due to DNA damage, due to protein damage, due to mutation, protein damage. And pathologic atrophy. Imagine this is a bile duct and there is a stone inside the bile duct, or outside there is a mass that gives pressure to this duct. The bile duct epithelial cells, they go for atrophy of the pathologic type. So this is another example for, and infections. This picture you see here is apoptosis. It's epidermis of the skin, and this single cell, you see the pycnosis of nucleus, the eosinophilic appearance of the cytoplasm is this cell that is apoptosis. Here it shows the apoptotic body formation separating from the nucleus. The mechanism of apoptosis that were intrinsic and extrinsic to form the apoptotic body. And the types of proteins, the BCL2, apoptotic, anti-apoptotic, and sensor, and the extrinsic or death receptor pathway. So, here, removal of dead cells, how the cells are removed. The first time I explained you about the cell membrane, plasma membrane, that they have some different names for phospholipids, including phosphatidylethanolamine, phosphatidylserine, phosphoinositol, and I said that usually phosphatidylserine is present on the inner side or outer side. Inner side, and phosphatidylinositol on the inner or outer, on both sides. Here in apoptosis, this phosphatidylserine that is present on the inner side of the plasma membrane, on the inner side. This is the lipid bilayer. In apoptosis, this phosphatidylserine flips out of the cell. So phosphatidylserine comes here. It flips out. And this flipping out makes sensing of the apoptotic bodies by the phagocytes. So this is one way that the dead cells are removed. How these dead cells should be sensed by phagocytes, by different receptors. So this phosphatidylserine that comes out of the outer surface of the plasma membrane, it is felt by phagocytes. Some other thing that is important for removing by phagocytes, that some of the apoptotic bodies have the thrombospondin molecule on them, and this can also be felt by phagocytes. So phagocytes can sense phosphatidylserine on the outer surface, can sense thrombospondin, and some apoptotic bodies also secrete some molecules that can be sensed by phagocytes. Also, phagocytes produce some proteins that it makes them way to apoptotic bodies. And again, there is covering of the apoptotic bodies by antibodies of the IgG type or complement of the C1q. Do you know the complement system? Complement system are proteins that can be both soluble or attached to cells. We have different systems for complement, the classical type, the alternate type. And one of them is C1q. This C1q or IgGs and immunoglobulin, they are bound to apoptotic bodies. And these can be attached to phagocytes. When a microbe or an organism is covered by complement C1q or antibody, this is called opsonization. In microbiology and immunology, opsonization is important for being sensed by our immune cells. If a microbe enters our body, then it is covered by immunoglobulin G or C1q, and then it is better sensed by our immune cells. This is called opsonization. This can also be seen for apoptotic bodies. They can be sensed by phagocytes, and so this is another type of removal of dead cells. So dead cells are removed through these things. Have I explained you about chaperones? No? In protein damage, the DNA damage and protein damage has one of the main causes of apoptosis. This is for protein damage. There are some proteins called chaperones, that these round molecules, they normally are attached to our protein polypeptides, and then they do the proper type of folding, because these proteins should have an accurate type of folding to have a proper function. So this is called folding. It folds normally to give its proper function. But if this protein is mutated, so chaperones are small molecules, they attach to this protein, chaperones, fold it properly. But if this protein itself is not normal, this folding is false, inaccurate, and it is called misfolding. So the protein, the folding is not correct, and this protein cannot function correctly. So one of the actions of chaperone is the accurate folding. And after this folding of protein, there are some other proteins called ubiquitin. When if there is accumulation of misfolded proteins, these proteins are attached to small molecules called ubiquitin, and then after attachment they enter a system called protosome that is composed of many proteins having protease action. These fragments come inside the protosome and are fragmented. So this is done on aged polypeptide chains, misfolded proteins, and if this action is done normally, the cell is healthy. So These chaperones make the proper type of folding. But if there is a stress to the protein, mutated due to UV, heat, free radical injury, anything, this protein is not folded like this. This is mature folded protein, but is folded improperly, we will have misfolded protein. If this system of ubiquitin and proteasome system functions correctly, the cell remains healthy. This reaction is called unfolded protein response. UPR is the unfolded protein response that normally this response is done on aged protein polypeptides and also misfolded ones that if it is done normally, the level of accumulated proteins is not very high, so the cell has the enough capacity to do this. So this fragmentation by the proteasome is done correctly, the cell remains healthy. This response is called unfolded protein response. But if the type of injury causes a lot of misfolded proteins, then there are not enough proteasome system to fragment this, then this unfolded protein response is not successful, and then we call, there is a stress to the cell called endoplasmic reticulum stress. In this endoplasmic reticulum stress, the cell misfolded proteins, abnormal proteins are accumulated inside the cell, and this is the basis of different diseases such as neurodegenerative diseases such as Alzheimer's disease. So the chaperones, ubiquitin, proteasome, the unfolded protein response reaction, if this is not normal, then we call endoplasmic reticulum stress, accumulation of misfolded proteins and diseases.
It's cause of the Alzheimer.
One of the examples, another is, for example, familial hypercholesterolemia. When the LDL receptor is mutated, LDL receptor for LDL type of cholesterol, it causes familial type of hypercholesterolemia. Another example is cystic fibrosis. In cystic fibrosis, there is mutation of the receptor of cystic fibrosis, which is cystic fibrosis CF membrane transporter receptor. This receptor is mutated. There will be accumulation of abnormal proteins. So this is the basis of some diseases, that some of them are common when we know them, and the mechanism is accumulation of abnormal proteins. And this, if these are accumulated, the cell goes for apoptosis. If the cell can do the apoptosis, we will see this type of cell death. But if the level of injury is very severe, as I said before, we will have necrosis. So always necrosis and apoptosis are not strictly separate from each other. Sometimes, so due to the level of injury, we will have necrosis or apoptosis. That's why we also have the term necroptosis that I will explain to you. Here we don't have necroptosis. I will explain it now to you. This is autophagia, is something else. So I explained you mechanisms of apoptosis and some examples, the system of protein fragmentation, accumulation of proteins. General pathology is not easy everybody should study well, but it's the basis of medicine. In any disease that you study, you will see that these changes occur in cells. So, apoptosis. Since all these are new terms, you should study it for not only one time. You should review it because there are new terms, many new terms that are similar to each other and necroptosis. Necroptosis is also called programmed necrosis. So this is called programmed necrosis. Also caspase independent type of necrosis, apoptosis. Caspase independent apoptosis. It's morphologically, it is similar to necrosis. So here we see cell swelling and plasma membrane degeneration, membranes degeneration, leaking of enzymes. But only it's programmed. Again, there are some genes that program this, but the features, the morphologic features, the biochemical changes are of the necrosis type. If we want to talk about its mechanism, there are some other kind of proteins here called RIPs, RIP1 and RIP2, that they are both attached to the inner membrane and also inside the cytoplasm. They can also bind caspase 8. So here the mechanism of apoptosis is this that we have proteins, receptors called RIP1 and RIP2.
It's for animal?
What?
It's for animal?
First, these are discovered in animals, but then it has the same name. If it's in human, it's different, it is mentioned. For example, caspase 10 is in humans, caspase 8. So this caspase 8 is an example in animals. So if we have this complex, if by this complex caspase 8 is prevented, so if caspase 8 is activated, then we will have apoptosis of the death receptor pathway. But if this complex RIP1 and RIP2 prevents this caspase 8, then whatever we see is the changes in necrosis. So this, if caspase 8 is prevented, then there will be lack of ATP generation and all the changes that we see in necrosis, and we will see necrotic changes. Calcium overload inside the cell, cell swelling, membrane degeneration. So this type of necrosis then it will be called necroptosis. So here we have caspase 8 if activated or prevented. Some examples are also are given for necroptosis. For example, growth of bone plate membrane. Physiologically, the death of cell during the growth of bone is due to necroptosis. But in pathology, acute pancreatitis, in reperfusion injury, in reperfusion type of injury, the type of cell death is necroptosis. In acute pancreatitis, type of cell death is necroptosis. And also in some infections, such as cytomegalovirus, CMV. In this infection, type of the cell death is necroptosis. So this is called programmed necrosis or caspase independent, because here caspase is prevented by RIP and there is no action of caspases. It's called caspase independent or programmed type of necrosis. So we see that its morphology is similar to necrosis. So this is necroptosis. Another type of cell death that we should mention, it's I think a previous of an edition of previous edition, but in the newer edition we have it, is pyroptosis. Pyroptosis means that cell death in infections. Pyro means fever. Again, pyroptosis is more similar to necrosis morphologically. Pyroptosis. In pyroptosis, this is seen in some infections that can activate another type of caspase called caspase 1. Another type of caspase is activated called caspase 1. And at last, what is activated in pyroptosis is interleukin 1. Interleukin 1 or IL-1 is the type of cytokine. Cytokines are small proteins. Interleukin 1 is type of a cytokine that is important in production of fever. So this caspase 1 activates first the pro-interleukin 1 and then interleukin 1 itself is activated, and this causes fever and the presence of caspase 1. And there is also another caspase here called caspase 11. These both caspases cause cell death through action of these caspases. And here again we have cell swelling. So cell swelling can be seen in necrosis, necroptosis and pyroptosis. Only it's not present in apoptosis where we have shrinkage. So again, the morphologic features are like necrosis, but the mechanism is different. That due to the action of some microbes, there is activation of this type of caspase, and at last interleukin-1 is produced. That causes fever, and caspase causes cell death. So this is another type of cell death: necrosis, apoptosis, necroptosis, and pyroptosis. When is the break time?
12:20.
So after break, we will continue. So next Friday.